All right, great. I'm sure a few more people will keep filtering in, and that's just fine. My name is Paul Jobsis, and this is Kristine Tartaglio. Used to be Kristine Funk. But Kristine has been working with marine mammals for quite a long time now, and been in Coral World since 2010. But her interest goes way back before then. Even when she was a kid, she knew she wanted to work with marine mammals. So that is pretty cool that she's got to live out that dream. And she's even worked in the same place where Jason and Dorian are, her other committee members, at the U.S. Navy Marine Mammal facility.
Intern.
And that was, and she was an intern there in 2008. Since being here at Coral World, she's been working with the South American sea lions, and that's been really great. I've known her probably since 2011 or 2012, and since then I've been saying, you really should come here and do a thesis. You really should come here and do a thesis. So this is a culmination of a lot of work and effort on her part. She started in 2017, which of course was a small little hurricane that came through here. Still get nervous when I talk about it. But many other changes have happened to Kristine since she started here. She's continues to get promoted at Coral World. So she is now the assistant curator for all marine mammals. She also, are you still doing the birds?
And the birds. Yeah.
And the birds, sea lions, dolphins. So she's responsible for a lot of stuff. There, she is also on the board of trustees for IMATA, which is the
International Marine Animal Trainers Association. Actually, just wrapped up my last board term with them.
So that is very impressive as well. She also got married during that time. Over here to Matt. Matt's very happy to have this come to a conclusion because he'd like to see his wife again. So anyway, with all that, I'd like to introduce you to Kristine Tartaglio, who will be presenting about alternating sound tasks to determine South American sea lions, audible, just noticeable differences in a marine park. I only got one more thing to say. I can't wrap it up. You guys need to know how special and difficult this is. You cannot just go out and say, I'm going to work with the sea lion today. So this takes a lot of work. A lot of things had to fall in place to do this. Permitting wise, just to even have a South American sea lion here is pretty unique. So this is very special that she was able to do this. So
Awesome.
Thank you
So much, Paul, and thank you so much, Dorian, Dr. Jobsis. Thank you so much, Dr. Jobsis, as well as Dr. Dorian Hauser and Dr. Jason Mulsow for being here all the way from California and for all of the expertise that you guys helped bring to this project. As Dr. Jobsis pointed out, this was a tremendous team effort. I had a lot of support to be able to accomplish this. So today I am very excited to be presenting to you guys my thesis using an alternating sound task to determine a South American sea lion's audible, just noticeable difference in a public marine park. I'm going to start with some background about my subject species, which was the South American sea lion, scientific name Otaria flavescens. This species has a very extensive range running all the way from northern Peru down the western coast of South America, wrapping around into the Atlantic Ocean as far north as southern Brazil, with colonies scattered throughout that range. They are classified as within the family Otariidae, and they, along with two other families, make up a suborder group of carnivorous marine mammals that is called the pinnipeds. Pinnipeds share the common feature of needing to be on land for an important part of their life cycle. And this is usually related to breeding, rest, and or thermal regulation. So because they tend to get a little confused, I'm gonna run back through those one more time so you really know exactly which group I'm talking about. The Otavenidae are walrus, the Phocidae are the true seals, and the Otariidae are the sea lions and the fur seals. And one of their distinguishing characteristics is that they have an external ear flap that you can very easily see. So that's who I'm going to be talking about. And the South American sea lion does share many of the traits that are common to the Otariid family. And these traits were recognized to favor individual recognition or recognition of another conspecific within your colony. And those include very dense populations within the colonies, a polygamous mating system, which does have an amount of aggression between individuals, especially during breeding season, and then required mother-pup separations, in which only a few days after the pups are born, mothers do have to go into the ocean to forage, and that can usually take up to 2 to 3 days, and then return to the beaches and need to reunite with their pups in order to be able to nurse. And so with all of these different requirements, obviously being able to find your pup, identify that male who beat you last week, things like that, could be very advantageous. And there are kind of 3 recognized ways that this is happening. One would be sight, one would be smell, and one would be sound. Now, it has been, and these are not necessarily, they're still working on necessarily proving which aspects they are using the most, and whether or not this is what really truly in identifying individuals. But there's a growing body of research to support that that's what's happening. And sound is the primary mode by which you would be identifying over a very long range. So I'm going to kind of go a little deeper into this, specifically about South American sea lions. What you're looking at is an example from the first vocal analysis of South American sea lions performed by Fernandez-Jurisic et al. In 1999. And what this is showing is the vocals just prior to a mothers and pups reuniting, and across the top, A through C is 3 individual different mothers, and G and H would be 2 individual pups. So this is a visual representation of that sound or the call that they are making just prior to being reunited. And to go into this, since we're going to be talking about sound kind of a lot here today, I want to go a little bit more into detail. So these bands of shaded area are representing a place where there is a more energy or a certain amount of energy of sound that's picked up in the recording, and indicating what frequency or pitch that sound would be. So when we say frequency, we're thinking tone. So in this example, that lighter with the red arrows, the darker band that's lower would indicate sound energy coming from a lower frequency, versus as you go upwards on that axis is going to indicate higher frequencies. And the darkness of the band, or the coloration of the shading, is indicating how much sound energy is coming at that frequency. And then as you go along, that's kind of what's happening over time. So there's a lot that goes into every individual vocalization, and just, you know, I think the untrained eye can kind of see there are similarities, but there are also some differences happening there. So what Fernandez-Jurisic et al. Did is they basically established 7 different parameters for which they could clearly measure within each sound, and then adapted and used models to be able to see if they could individually identify each vocalization with the individual who made it. And for 5 of those 7 parameters, they were able to do that at a 90% correct rate for the females. So there was a lot of other things they found there, but essentially what they discovered was the South American sea lion does have a lot of vocal variability, and likely does support the possibility that you could recognize individuals on it. Now, the other part of being able to use sound is being able to hear it on the other end. So some of the common methods that are being used in assessing marine mammal hearing right now are vocal analysis, like the one that I just showed you, where you're looking at the typical range that the species are producing vocals at, and making an assumption that they probably hear most of those. Also looking at the inner ear anatomy, so through necropsies or other modes, being able to look at the shape, size, and kind of characteristics of the inner ear, and comparing those to other species that we know how they conduct different sounds. And then the last one is a direct hearing assessment, which is one of the most ideal measurements. Obviously, all data is good, but being able to directly measure how well an individual within a species hears is the ultimate kind of information that we want to know. So there are 2 primary ways that this is being done. One would be called through using psychoacoustic or behavioral tasks. And so that would involve something that probably many of you are familiar with, where you would participate in the hearing study yourself, or in an assessment wearing headphones, and you listen, and when you hear the sound, you raise your hand or you push something to indicate that you've heard that sound. Does that sound familiar? Yep. So that's the behavioral or psychoacoustic assessment. There's also an electrophysiological assessment in which they are measuring the electrical pulses within the brain that are being conveyed in response to hearing different sound waves. So those are the two ways that are contributing data currently. And Malsau and Reichmuth in 2010 performed a comparison of psychoacoustic hearing assessment to electrophysiological assessments, and did find them to be approximations to each other. However, the psychoacoustic tests are routinely, especially the lower frequencies, showing the lowest sensitivity rate. So that is kind of remained, the behavioral assessments have remained sort of the gold standard measurement within this research. So previously there was no direct hearing assessments for any otariid species in the Southern Hemisphere that I could find. And so with this data, what is being done with it is pretty interesting. And this is really taking a lot of small pieces and putting them together. I wanted to highlight one specific example that's really very impressive, which is which was a publication by Southall et al. In 2007, which basically compiled all of this data, all of the hearing assessment data for all marine mammals that they could find, and then kind of filtered it into using with a certain set of criteria for how it was collected, and then took all of that compiled data and was able to group all marine mammal species based on their predicted similarities in how they predicted they would hear based on all of that data. Once they made these groups called marine mammal groups, then all of the species within every group they were able to combine all of the data and start applying it and applying it to what's called a weighting function. And these were originally developed for humans in order to help reliably predict how well or how a person's ear was going to interpret different sounds. And it was later adapted to also be used to predict how human ears, or the normal human ear, might be affected by long exposures to loud sounds or loud sounds or anything like that, and whether that would cause any hearing loss or damage. So they used this function and adapted it to marine mammals. So they called it that M weighting functions, and they were able to do that for all of the marine mammal groups and provide a very useful tool that could be used for regulation and protection of marine mammals around the world from adverse effects of being exposed to different noise levels. So for those of you who aren't as familiar with how marine mammal data is collected, Paul alluded to it a little bit, but it is not easy to get, but it is really important. And so being able to combine a lot of data is really what this example did. And so in 2019, Southall et al., that publication was updated, and this document was 107 pages long. So I didn't read all of it. I did read the most important, the section relevant to me, which was the group that was called Other Marine Carnivores in Air. And so the entire Otariid family does fall within that group. So that's the one I'm going to be specifically talking about today. I took data from their appendices and made this graph to show you where how many behavioral assessments of hearing contributed for each marine mammal group. There is one marine mammal group that is missing, that is the mysticytes, for which there is no behavioral data available. But for the other marine carnivores, there were seven assessments included that represent that group, and the mean subject per study or per publication within that data was 1.1 subjects. So, and actually some of those subjects were repeatedly measured. So it just goes to show how different marine mammal research can be from some of the other conservation research that we might be used to. But oh, is there oh, that's I don't know, probably a good note. We don't want it turning off. I don't know which button to push. Okay, are we good? We'll see if we lose it or not. Okay, all right, there we go. So All right, so there is a lot that we know. There's, of course, always a lot that we don't know. And frequently within the literature, it is commonly referenced that just adding more data for more species is always valuable. They are a very, very diverse group of animals. And so getting hearing assessments on more animals and novel species would be very important. And as I mentioned, the South American sea lion, not only the South American sea lion, but none of the sea lions or odoriid species in the Southern Hemisphere have direct data. Also, measuring and assessing novel aspects of hearing. So I mentioned that the Jurassic and the Fernandez and Jurassic assessment, they identified and they measured 7 different aspects or acoustic traits of those sounds. So there's a lot of different things that go into hearing any given sound and what makes up that sound and how it's perceived by any individual. So one of a measurement that's a little bit different than what I've been talking about so far is called a just noticeable difference. And so this, what I've been talking about already was absolute threshold. So those hearing studies that you guys have participated in, they're very simple. They're usually a single tone, and you're looking at how quiet can that get and before you don't hear it anymore. And it's also called an audiogram. So this is what we do have quite a bit of data for some otariid species. And this is a representation of 3 of the most genetically distinct species within the otariid family: the northern fur seal, which is represented twice here, the California sea lion, and the Steller sea lion. And you can see pretty clearly that they are similar. And that was something that Malsaw and Reichmuth in 2010 used to form the hypothesis that within the otariid family, very likely those species are all going to form a functional hearing group in which we would expect they probably hear very similar. So even though the South American sea lion hasn't been formally measured, it does fall within this group, and we might predict that it falls similarly. So what you're looking at here again, and just real quick to review those: frequency, you're thinking tone. So from left to right, these are going lower frequencies to higher frequencies. And then that is measured in how loud that sound needs to be able to be perceived reliably. And so these were psychoacoustic tasks that were produced where a sea lion actually, or a fur seal, responded repeatedly to report when they heard something or didn't. And these are essentially called a threshold task. And you can kind of see there that on the low end and the high frequency end, that those needed to be pretty loud. And then around 10 to 12 kilohertz, those are very quiet sounds that they were reported that they could hear. So to get into just noticeable difference and what that exactly means, I'm going to do a couple of examples, but essentially a just noticeable difference could measure anything that you can perceive. So from the brightness of one light bulb to another light bulb, or if you walk into a room and the presence of a smell versus another room, all of those things could be measured. But what I'm going to be talking about is sound. And I'm going to be looking at the smallest perceivable difference between 2 sounds, specifically volume. So everything else about these sounds will be the same, but at some point the volume will be different. And we're looking at the place where he can no longer, or a sea lion can no longer tell that those 2 sounds are different anymore. So as an example, and I apologize, if anyone here has some high frequency hearing loss, you might not be able to participate in my examples fully because this is an 8 kilohertz tone. But if you hear this sound, many of us in the room, okay, good, we got we can all pretty easily agree that it's a different volume than this sound. But if we hear these 2, they're close, or maybe they're not. It gets a little bit harder, right? So that's kind of the idea. And the way that this has been scientifically investigated or assessed is using kind of a standardized task. So it might sound something like this. And I'm going to have everybody participate on this one, listening, and raise your hand when you think you hear the volume of the tone change. All right, cool. So a lot of people eventually heard it. There was some differences in, some people didn't. That's okay. A lot of people when I practiced didn't hear it, so don't worry, you guys aren't alone. So that's basically the idea of what just noticeable difference is. Now, when you are when you are trying to figure out a threshold or a minimum, this is the same with absolute thresholds, those audiograms, but also the same with finding a just noticeable difference. You need to make sure that you are using a standardized method. And so there are a lot of methods that have been previously validated for comparison and offer different advantages or disadvantages. I chose to look at these three specifically in my environment and in my context. So starting with what's called the adaptive descending staircase, and using this method, it adjusts the difference level, so from larger to smaller, based on the response of the subject. So if you look at my kind of informal graph here, as you would go down on this graph, that's indicating a smaller difference that's being presented and asked to be basically that trial of do you hear this or not. And then across each little circle indicates being asked again and again at those different levels. And the green would indicate that they reported, yep, I heard that difference, I heard that difference. Red would indicate they did not hear that difference. So the idea is then with this schedule, you would bring it a little bit up, and you would start at a predetermined level and decrease by a systematic procedure, and then based on that response. And eventually, ideally, you get to a place where you can go up one kind of level and they hear it, and you can go down and they don't hear that. You go up, they hear it, you go down, they don't hear that. And that's kind of how that works. The method of constants would basically take all of the difference levels that you wanted to assess, put them into an Excel spreadsheet and just mix them up, randomly generate that, that session, and just ask the trials sequentially to the subject, record the answers, and then you can analyze that data afterwards for which ones they were hearing and not hearing. And then the third one is called the repeated descending staircase. And similar to the adaptive descending staircase, it starts at a pre-established set point and then follows a standard systematic decreasing based on the subject reporting that they do hear that difference. However, after the first time that they report, I don't hear that, it resets back kind of at the top and goes back down. And so the adaptive descending staircase and the method of constants are probably the most common that I saw in the literature for threshold detection tasks, especially in research facilities. However, I noticed that the repeated descending staircase was used in a facility that was much more comparable to the one that I was planning to conduct my research in at two different aquariums. And that was by Reichmuth et al. In 2020 when they assessed an audiogram for walrus. So the previous Otaria just noticeable difference measurements that have been done, there are two of them currently. And the first one was done by Moore and Schusterman in 1976. This was performed actually underwater, and the subject was an 11 to 12-year-old male California sea lion. They used a block method descending staircase, and the tone frequency was 16 kilohertz that they tested. And the just noticeable difference that they found was 3 decibels different. The other one was Mulsau and Finneran in 2011. This was presented in air to a 2.5-year-old male California sea lion using an adaptive descending staircase. They used an 8 kilohertz tone, very similar to the ones that you've heard. And then they determined something less than a 2 decibel difference. However, that data was not officially finalized to the final just noticeable difference. So based on this information, my null hypothesis for 8 kilohertz pure tone stimulus pairs presented in air was that a South American sea lion's auditory just noticeable difference would be 3 decibels difference when measured as the smallest difference detected significantly greater than 50% of the total trials attempted. 50% is a relatively common set point within signal detection research. And I will be evaluating gathering this data using the adaptive descending staircase, the method of constants, and the repeated descending staircase. Not necessarily making a comparison of those methods because that has been done, but evaluating using them within the context of a public marine park as opposed to another facility. All right, getting into the methods. So this entire study did take place at Coral World Ocean Park on St. Thomas in the US Virgin Islands, and the data was collected within our sea lion area in the back. Our subject was the one and only Romulo. He, at time of data collection, was a 17-year-old neutered male. still is neutered male, as a South American sea lion. And he, prior to embarking upon this, had no previous experience with any kind of threshold testing paradigms at all. So he did have an extensive history with operant conditioning, in which he had a lot of foundational behaviors we used in order to train this behavior, but he had never done it before. He was selected based on his availability for sessions, his comparability to previous assessments, as well as the potential for future replicates at Coral World. And all of these methods were reviewed and approved by Coral World's Institutional Animal Care and Use Committee. So I should say the final setup, we went through a lot of revisions, but in the end, it looked like this. All of the data collection took place in essentially the hallway that I converted into my study space. And on the left here, the panel A shows Romulo at his station point with his trainer, Gabby, who is present through each of the trials and available to provide the reinforcement and adjust the headphones as needed. Behind them is the white door, and I am on the other side with a computer that's able to control all of the parameters of all of the trials done throughout the process. And then the C panel is showing you the view from the live stream video that was allowed the researcher to see Romulo as well as the response target that's just to the left of him. You can see it's a little white square, although actually he broke that halfway through. So halfway it just changed to a target. So they are different in these pictures. All right, so that's what it looked like. The behavior, so I've talked about the task that allows us to assess this is called an alternating sound task. And there are two different potential trial types that he would have to learn how to answer. One would be a change present trial. And this is one in which there was a change. And then the other one was a change absent trial, also called a catch trial, in which there would be no change in the volume throughout that entire trial. So when the headphones went on, it would be pulsing like the example you guys heard. Then at some point, the trial would start as soon as he's on the chin station, and that would initiate what we call a variable delay period, and that would be anywhere randomly from 3 to 10 tones, and that was all randomly generated by the end. And then that initiated what we call the response window, which was 5 alternations of that softer and louder tone, which gives the time where Romulo has the opportunity to respond and say, I heard that. The catch trial, all the same format, with the exception of the response window, continues to pulse the same volume. It doesn't change. So for Romulo, what he is supposed to do, or basically what he does in response to, in the response window for a change present trial, if he hears that difference, he can report it by picking up his chin, moving to his left, and touching the response target. Alternatively, if he does not hear a change, then he should remain on the chin station, basically waiting still to hear it. However, if it is the end of a change present trial and he's still chilling on the chin station, hanging out there, that would be incorrect. He essentially missed that. And if he, during a catch trial, does turn to touch, that would be labeled a false alarm, also is a correct response. False alarms not only could happen at any point during a catch trial, but that could also happen during the variable delay of a change present trial. And the catch trial, the goal there is to create a balance. So there was a reward option for not guessing, as well as for detecting that tone difference. So for all correct responses, he received the word good from the trainer and a fish of equal value. For incorrect behaviors, he received just a brief 2 to 3 second pause, and then we reset and moved on to the next try. The control panel was a customized software program built in LabVIEW and used by National Instruments. Then this was pretty cool. I have to thank Dr. Jason Mulsow for actually coming to St. Thomas and helping me get a lot of this set up, learning how to use it. But once we got it running, this worked really great. I was able to manually control all the parameters of the test. So in this example, you would be seeing basically that A tone would be a 60 dB sound. SPL is another measurement for that volume. And then 68 would be the B tone, where this would make an 8 decibel difference sound. I could also control whether it was a catch trial or a change trial, and when the trial started and when that change was initiated. So when you put it all together, it looks like this. This is, again, I apologize, an 8 kilohertz tone. And it is the view from the researcher's perspective, but you would be hearing what Romulo would be hearing. No one else would be hearing this.
Good. Nice job, bud.
And then the next one is a change trial.
Good. Good.
When he was sure, he was very sure. All right, so I did mention he did not know how to do this when we embarked upon this project. So we started with training, and also neither did any of the animal welfare specialists at Coral World or myself. So we had a lot of learning to do. We started together all in a room so that we could communicate, knowing this was not a double-blind condition, but essentially trying to figure out how to train this behavior. So I think if we did it again, we could do it a lot faster. And essentially the goal here was to keep his motivation consistent, to get him to learn every part of the trial, every part of the task, and then make it more and more random without making it too hard. So we had to move at just the right pace for him, and then eventually give away all control to a randomized program that would determine every part about the trial. At the beginning, I was able to kind of decide, we got a feel for which trials would be a little easier and which ones would be a little harder. And we kind of carefully programmed that to get him to be able to do everything. So once we felt like he was ready, we would move into a learning effect test. And essentially this was a double-blind condition in which Gabby, who was with him, did not know anything about which trials were coming, other than there were going to be 15 predetermined trials that were a random mix of change trials and catch trials. And then those would be about an 8 to a 10 decibel difference range, so that based on previous assessments, we felt confident he should be able to hear those. So he had a good chance to get a high portion correct here, and that was a requirement. So he needed to get above 90% correct, as well as a lower than 30% false alarm rate. And he needed to do that for 4 trials in a row before we were able to move on to data collection. At any point between these two, we did do a little bit of back and forth to essentially get him to the point where we felt really good, and his results showed that he knew this test. Once we moved into the just noticeable difference testing session for each method, we did fully complete that data collection before moving on or coming back to any kind of training. So this was also a double-blind condition, and as I mentioned before, the parameters were going to be determined by the method that was being evaluated. So for the adaptive descending staircase, for each individual session, our goal was to get 5 sort of 1 up and then 1 down of those reversal patterns before we would end that session for the day. The method of constants would be to complete all of the randomized trials that we wanted to assess that day, and it was about 30. And then the repeated descending staircase would be to complete the desired number of transitions. Again, we wanted to see consistent motivation, so he's participating consistently, a false alarm rate less than 30%, and consistent background noise that both the researcher and the trainer felt were not inhibiting or affecting his responses. So there were occasionally times where we had to, because of things that were outside of our control, the power went off on us one time and the generators fired up while we were in this hallway with him. So we did have to terminate those sessions, and then those data were not used. So for anything with when you're measuring sound, you do want to make sure you're measuring everything. We did a 1-minute sample before every single session, and I was able to use an external calibrated microphone that plugged right into my cell phone, downloaded an app, and was able to get an assessment of the sound in the room just prior to that test. I was also able to frequently do a calibration check on the headphones to make sure that the sounds I thought I was producing based using the computer were in fact the sounds that were being transmitted to Romulo's ear. And what we got for those, this is the results from all of those measurements, were basically what we expected. We knew there was going to be a lot of background noise. We did have a reverse osmosis machine and some filtration pumps running. So the orange bars here are showing a broadband spectrum of frequencies. So this is including a lot of low frequency sounds as well as high frequency sounds. The blue bar indicates just a 1/3 octave band around the 8 kilohertz frequency, which was the tone that he was listening to. So that one we were glad was quite a bit lower. The good part about all of these measurements is the variability was low. So the comparison between each different trial type and each different individual session, he was in essentially the same conditions every time. And then again, the measurements for the headphones showed no significant difference between any of the positions measured for either of the headphones. And then lastly, how we determined what the overall just noticeable difference would be for each method. So in order to do this, I took just a snippet of my data. This is actually what we got for 5 dB difference for the adaptive descending staircase. For that level, the total number of times he attempted that difference level was 23 times. And out of 23 times, he positively detected that 18 times. So since we wanted to determine, was this significantly greater than 50% correct, we used a one-sided Wilson score test with continuity correction set at a 0.05 value to generate confidence intervals, as well as a p-value to determine if that was significantly greater than 50% based on binomial proportions. And this was specifically chosen because we knew we were going to have a relatively lower sample size. And false alarm rates were calculated for every single session. All right, getting into what we found. So I decided to put up all of the raw data for the learning effects test since there wasn't too much going on there. These are what he did for all 4 of those 4 in a row trials or sessions. And we did end up deciding to run a second learning effects trial because due to scheduling, we had to take about a 3-week break before we evaluated the last method. So since we were coming back, I wanted to make sure that nothing had changed, and we did find very quickly that he picked it right back up, and the correct detection rates and the false alarm rates from both of those were not found to be significantly different than each other when compared using a t-test. And for the official just noticeable difference results, so this graph is showing you the general trend that you can see for all 3 different methods assessed. As we move from a ½ decibel up to 8 decibels difference, you can see this percent correct going up. And overall false alarm rate for all 3 of those methods were well within the range that we wanted, so less than 30% and greater than 0. And they were all not, when analyzed with an ANOVA, they were found to be not significantly different from each other. This is good because I wanted to be able to show that he was performing this test consistently. So basically, this is kind of how conservative his responses were, or how often he was guessing. And so it is a good thing that these were all the same. And so then when you kind of look back at each methods and you compare them just to a straight 50% comparison point without adding any statistical analysis, there's an interesting pattern going on with the adaptive descending staircase and the method of constants in the light blue and the dark blue, which actually dip below 50% and just following 4 dB difference and 3 dB difference, but then he had higher than 50% correct at 1 dB difference. So this is interesting, and with Mulsow and Finneran's assessment in 2011 being something less than 2, I think that there's probably, this is kind of just really makes me curious to want to do a little bit more and see what's going on there. However, when we do add in the analysis to be significantly greater than 50%, we determined for the adaptive descending staircase that his just noticeable difference was 5 at a 0.006 level. And for the method of constants, it was 4. And for the repeated descending staircase, it was also 4. So based on these results, for all 3 of the methods that were evaluated, I'm rejecting the null hypothesis in favor of the alternative that a South American sea lion's auditory just noticeable difference does not equal 3 decibel difference. So moving into the scientific contributions to be discussed from this project, we did successfully collect auditory just noticeable differences for a South American sea lion. This is the first direct hearing assessment for a South American sea lion. And I like telling our guests who visit that we probably, as far as I know, we have the only South American sea lion in the world who can perform a psychoacoustic task here on St. Thomas at Coral World. So that is our little claim to fame right now. And then also we have further adapted and evaluated the threshold assessment methods for being applied in a marine park and for gathering just noticeable difference data with an Otariid species. So a brief kind of discussion of the methods and our findings there. So for the adaptive descending staircase, what we found, this was evaluated first, and we found challenges in obtaining all 5 reversals. So as what he would get there really quickly, but then go up really high, and we had to end all of those sessions before we were able to obtain those reversals for other reasons, running out of time or sound or any or a change in motivation. For the method of constants, this is known to be a highly resource intensive method to perform correctly. And I had saw it in the research and I really wanted to evaluate it and try it. But we did discontinue this method after only 3 sessions based on the decline that we saw in his motivation and a little bit of increased frustration with it being probably a little too challenging based on the balance of hard near threshold tasks versus the easier ones, right? So if it's every repeated trial is a little bit too hard, that's not going to remain being very much fun to perform. For the repeated descending staircase, this was the 3rd method evaluated, so I think that's important to recognize. However, we were able to consistently achieve the desired transitions daily. Nice thing about this method is that it was, which I was hoping for with the method of constants but didn't work, is that the session duration was predictable. And so we did kind of know what we were going into with, if we had a certain amount of time, we could get an idea of how we knew exactly what we needed to achieve. And overall, Romulo's motivation stayed pretty high for this one. So, all right, so another thing that I mentioned that we monitored the background noise in the space, in our testing space, and we knew this was going to be one of the biggest challenges as compared to facilities that have rooms that are designed and set up to mitigate noise. This is really hard to do. I have a new appreciation for how noisy our world is. Oh, actually, I forgot to tell you the official number. So for the 8 kilohertz, the 1/3 octave around 8 kilohertz, the testing level in my room was 52 decibels. So, and using this representation from Malsau 2011, which was a measurement of the sound in the testing space that was used by Malsau and Finneran in 2011, they found about 14 decibels for that 1/3 octave band around 8 kilohertz. So that is a really big difference. Although with where we felt that we were presenting these tones to Romulo, and if you account for the fact that once you put the headphones on, that also reduced some of the ambient noise that was at his ear level, we do feel confident that the actual tones he was hearing weren't impacted or masked or covered up. So we should have been able to get a good assessment there. But when you are thinking about making comparisons in any way, this is important to know. Also, when you are doing single subject studies, we want to talk about the characteristics of that individual. Romulo was 17 years old, so at this point, the oldest South American, or the oldest Otariad to be assessed for just noticeable difference. And there is, of course, no previous information for Otariads and age and how just noticeable difference might change, but there is some for humans. Freegang et al. In 2011 did find that in older humans did have a significantly higher just noticeable difference than younger humans did. Schusterman et al. In 2002 documented for an absolute thresholds, and so testing an absolute threshold, what we were talking about at the beginning, they actually showed for a California sea lion that there really wasn't much notable loss beyond about 20 years of age. So I think there's a lot of interesting things that could be looked into there. And also I mentioned, oh, there was my birthday pic. So he was 17. I missed that one. Learning history. This is what Romulo is a little bit more familiar with prior to doing this task. I think learning history is important, and then probably also cueing into methods. So if I was going to start again from scratch, that repeated descending staircase felt much more achievable. Although at this point, Romulo now does have experience with psychoacoustic testing with that paradigm. And so he is an animal that we could potentially be able to do and move forward with, who does have experience with this task. But at the time of data collection, that could have been a factor that was different. The previous California sea lions that were assessed were known to have previous experience with this type of test. And then reproductive status, I mentioned he is a neutered male. The intact males in the South American sea lion species do look notably different. And actually even their ear bones have been noted to be much larger than their tympanic membrane, their eardrum, larger than juveniles and females. So I think if when you're talking about the species and the assessments, that's also important. And when we bring this back and start thinking about wild populations and what's going on with the South American sea lions on the coast of South America, they're overall known, they're stable. They do have a stable population, and they are not recognized as a threatened species. They are a species of least concern by the IUCN Red List. However, within individual colonies, there are fluctuations, and some of them they have attributed reasons behind, and others are fluctuating or declining, and they're not necessarily able to know what's going on. But this one was one that was done by Túnez et al. In 2008, and they assessed a large list of different factors for association with number of breeding colonies, or with the presence of breeding colonies and the abundance of individual sea lions within those colonies. And they only found 3 that were significant. And the only one that was positive was islands. So islands was positively affiliated with breeding colonies being there and a higher abundance of sea lions within those colonies. The other 2 may not surprise anybody, but those were the presence. They were negatively associated with the presence of cities and increased populations. So there are some documented anthropogenic influences going on there. Another study in a different location, Dans et al. In 2004 assessed 4 different colonies over about a 20-year span, and they documented almost a double in population over that time. And they measured the breakdown. So what you're looking at is actually the breakdown of the proportions of different groups of individuals within that population as it grew. And what the big one that he points out is juveniles, and the proportion of juveniles increasing within that population, and pointing to pup survival as a key factor that might have been driving that increase. He also pointed out with the increase in population, new breeding colonies were formed outside of the original research locations, and that many of those new colonies were not under the same within marine protected areas as the original colonies. So there are a lot of impacts out there that are happening, and I think that with the unseen effects of noise pollution and the lacking understanding of behind just how and what these animals are able to hear, that some of those impacts could potentially be magnified. Also, the other thing in reference to their population stability and the importance of being able to, and how important it is to contribute to the management of the species, was pointed out by Oliveira in 2011, in that they documented this image is showing a breeding colony in the year prior to one of the most significant El Nino events in 1997 and 98. Their breeding season corresponds kind of very much right around December, January to when that was happening. And so you can see the difference between before, and then the B panel is showing you after, and very much linked to the decreases in prey availability. During that year, there was a 100% mortality rate of pups born. There was a very low rate of pups born, higher rate of abortions, and this also impacted when pups were born in subsequent years. So with the predicted increased frequency of these events happening and having such drastic impacts on some of these individual colony populations, the argument was really made that more needs to be known about the species and about being able to support the population growing in between these different years. So with that, some of the future directions that this could take and that I would recommend would be just increased trials. So further running or narrowing down with the methods, and then being able to further run more repeated trials to give that the statistical analysis some more power behind that, as well as replicate subjects. So, and including in those females, juveniles, and intact males. The other thing that I would definitely want to highlight is that despite all of the challenges and the limitations, the partnerships that make this possible are critical, and they between the work that was done collaboratively with zoological institutions, with Coral World, with the National Marine Mammal Foundation, and with UBI is really what allowed us to be able to make this happen and be able to create an increased opportunity for more data to be obtained. The Coral World is actually one of only 2 facilities in the United States that has South American sea lions living with them. So we were really excited to be able to contribute information about them. Also, as I kind of put my curator hat on, I guess, a little bit here, but with my experience working with marine mammals and with behavior, the act of just learning something new, when it's done well, learning and just doing that behavior can be really rewarding and can be very enhancing. And so I was really excited to bring this as a brand new, very different kind of learning challenge to our sea lions at Coral World. And we did see him he gets pretty engaged in it. So that was really fun to see, as well as the staff experience. So the staff at these institutions, both from the academic side, being able to see what goes into animal care and learning about them, but also the staff at Coral World being able to get experience assisting with a scientific study and performing the training for this task. When I reached out through the International Marine Mammal Trainers Association, I got looking for people who had trained this, I got pretty much nothing. No one had ever trained this that I was talking to outside of the facility really where Dorian and Jason were working. So getting that experience was very cool. And then the built-in education and outreach component that is just there at the zoological institutions. And we were able to piggyback off of Coral Worlds, and Coral World was able to use this. So as of April, so this is actually the numbers roughly for just 2022 alone, based on our booking system. We have had roughly 3,000 people walk through this study space and learn about this research and be able to take a little tidbit or something about it, and then possibly even go meet Romulo and take a picture with him. And I've even seen a few TripAdvisor reviews now that say and recognize the cool research that's being done. And then I met the sea lions, and it was really, really neat. So just all of those benefits are huge, and go even beyond that, increasing the data that is brought back out of it. So with that, I'm going to leave you guys with a little clip of this. Was actually the very first time that we at Romulo took a cue from a sound that neither Gabby or I could hear. So the first one, we actually give him the answer.
Good.
And we're gonna wait and see what happens on this one. So that actually was shared on Coral World's social media page, and within just a handful of days was shared almost 100 times and had over, was it 1,000 likes? Yeah. So again, mutual benefits there for both facilities were present. So with that, I have to acknowledge so many people and facilities, which I've kind of already done, but Coral World, Trudy and Neil Pryor, as well as Lee Keller, could absolutely not have done this with your support and your backing. The Sea Lion Animal Care team was amazing every single day. To be able to get these in over and over again was not easy, and I could not have done it without them. Gabrielle Gamage, who is leading that team and also helped me on a lot of her days off, as well as my volunteers, the personal support that got me through, and my husband, Matt Tartaglio, my committee members, again, Dr. Jobsis, Dr. Malsau, and Dr. Hauser. They were great and continue to be great, even though they got a little more than they signed up for, how long this took me. And the financial resources and support, again, from Coral World, the National Science Foundation, and the Lana Vento Scholarship Fund, all helped contribute to making this possible. So with that, I will be happy to take anybody's questions. Neil has one.
Is your experience with the accuracy of your sea lions similar to the accuracy obtained by those other studies of different sea lions?
So that is a good question. In a lot of the publications, they don't show all of the data. There's a lot of data, right? And that was one of the things I recognized as we started. I do think that because Romulo had never done this kind of task before, it was not as good. And I subjectively felt like he got a little better as we continued to, and especially when it got hard. And it's different individuals are different, right? Like, you may really like to do one kind of activity and somebody else may hate it. And I find the animals seem a little similar. So we did find something true to Romulo that we already kind of knew about Romulo is getting things wrong. And when things get a little bit hard, and kind of working him through not quitting or giving up on that was something that we did have to work on. Yeah. Yeah. Did you have one, Amber?
So I'm gonna ask you to anthropomorphize a little bit.
Okay.
And you kind of touched on this, but do you think that he enjoyed this work?
That's a
And
good
if you
question.
could say, like, if you don't continue with it, do you think he's gonna miss it?
I didn't thought that far. Well, I'll start with your first part of your question, which is, do I think he enjoyed it? And I think I do. I do think so. And especially at the beginning, of course, as it got a little harder, there were days where I feel like there were days where he was like, not so much. And that was where I mentioned the motivation. And I think it could be a lot of different factors. And Gabby and I were always trying to figure out, like, the behavioral side of this was really interesting because we had to gain his participation. But one thing we noticed at the beginning, early on, that the testing space was in a hallway, and Romulo had to go through this hallway to get to our presentation space. So we do these educational presentations, and the sea lions walk out into the aquarium to do them. And I noticed as he walked out, he was looking, really normal and walking a normal pace. And then we went through the testing. I was like, I wonder if he thinks we're doing research, but I knew we weren't. We were going out to do the show. And once we got, and I just noticed a little decrease in the energy, a little decrease in the focus once we got out to do the talk that we, have been doing for years. And I thought, it seemed to me anthropomorphically that he was almost disappointed. Like there was this shift in energy when it was like, oh, we're not doing the research. So I kind of thought that was really, really cool. And just, yeah, like when he started to figure it out, his energy was fun to watch. Yeah. Good question.
I have a question. So I'm going to start with a statement first. So I started off talking to Kristine about doing this as like a electrophysical measurement. So basically using the electrodes. I thought that will be easy.
It's like, really? You can just do that? That sounds great.
And then Kristine educated me and said, that's really not the best way to do it. Really have to do it behaviorally. So I was all on board. It was amazing what Kristine and her colleagues can teach the animals to do and not do and all that. So you got to do this trick. But then my question is, when you went to present this at IMATA, you got some advice about what type of research you should or shouldn't do in a marine park. So what type of what advice did you get?
Yeah, so I didn't present it, but I did attend a forum on research, talking about research training and conducting research at zoos and aquariums. And one of the panel members, afterwards I ran into at an event, and so he had mentioned, they were talking, the question was asked, where would you start? How would you start? A lot of the animal care, the people working with these animals, we'd love to know more about them. We know that there's things that we could be doing, and we would, willingly give our time to help with these projects, but where do you start? And his advice was probably good, and maybe had I had it 7 years ago, this would have been different. But he said, start with something that you have. He said, I wouldn't recommend that you go and dabble in bioacoustics research, which is exactly what I did. I was like, something. I was like, oh, I think I can affiliate with that more than almost anyone else. That is really what I did. I dabbled in bioacoustics, and I underestimated how complicated just sound. I thought the training and the aspect of all of that and scheduling, but just understanding sound and being able to measure it and trying to control it, and that was a whole nother area that was super interesting. And I'm really glad I have that kind of base knowledge for it now, but there's a lot out there about that. Yeah.
So I guess kind of following on that, it makes me curious too. Did you take any lessons from your results here and think about how the animals are being cared for at Echo? Like you mentioned, you measure that sound quality. Like, are there concerns about how loud it is there?
That's a good question. So that is in, maybe more for the staff than the sea lions, that's in the office area. So yeah, actually, I was like, oh, it is quite, we always knew it was a little loud, but now I have a number. And so there's been talks about taking down my sound. I made some attempts at mitigating the sound using, it's called mass loaded vinyl. So I ordered this super heavy stuff that I lined the walls with. To be honest, it didn't work very well. And I think my committee knew it wasn't going to, but I tried, and it looked cool. And then basically trying to isolate some of that sound. So yeah, it's been talked about, but yeah, it is more in the building, so it's not necessarily affecting the sea lions. But one thing that we have talked about is using this as an assessment that maybe we would do annually if all of the sea lions learned this and we were able to monitor and see, is there, because we do use our sound to communicate with them a lot. And we talk to them, we know they hear our voice, and they take cues of different inflections. So I think it would be kind of interesting to know. And one of our sea lions at Four World does have ear infections. And we knew this was part of actually kind of partially also what interested me in looking at hearing. And so maybe being able to see, monitor him for any changes going forward as well. Yeah, it's a good question. I forgot to repeat that question. Oh, I have another one. Yes.
First of all, congratulations.
Thank
so
you.
Happy to be here. I remember when we first started talking about this, and I was Yeah. Mind blown, mind still blown. So I guess following up on Joe's, with the care specifically for the sea lions, do you think that this data could push forward into, the dangerous zones of sound level for other wildlife here in the Virgin Islands?
I'm sorry, maybe rephrase that. That's a good, wow, that's a good one.
Yeah, it's a big question.
Yeah.
Sorry.
One more time.
So,
You want
how
another?
can we take studies like this to have sound control that benefits more wildlife in the Virgin Islands other than just the sea lions?
Yeah. Okay. So how can we taking this concept and moving it forward to benefit other wildlife in the Virgin Islands? That's a good question. So something that I think the big thing for me is understanding the ambient noise around us, understanding and being able to even assess, where are there assessments? One that we've talked about a lot at Core World is Water Bay, and what is the sound that's going on underwater? And because we do know, and then which species do we know that rely on, or we think that they rely on communication, and where are they doing it? And so I think just pinpointing those to start with, and then being able to set up acoustic monitoring stations and understand what is the baselines would be a great place to start. Using some of those, if you don't have an animal living with you in human care who you can train to do these tasks, you pretty much have, looking at the anatomy of their ear, any previous published research, or the vocalizations that they make, are they within the frequency ranges that say the new, windmill that's getting put up somewhere is going to be producing? Is that going to be inhibiting or interfering with their communication? So yeah, I think that's maybe not necessarily for just noticeable difference specifically, but yeah, I think the fact that we've gained this kind of appreciation for that would be huge. I'm excited to start applying a little bit of what I've learned and possibly have more collaborators looking at sound in Water Bay for sure. Yeah.
I don't see Sophia here.
She had to cut out. I knew she was going to have to.
So I don't know if there's anybody on Zoom
should I
that
check?
has questions, but she was monitoring that before.
Okay, let's see. All right, let's see. I see a handful of people. I don't know.
There's some things on chat there. That we could.
Oh, there we go.
Maybe I was supposed to tell people to do the surveys.
Okay.
So do the surveys.
Please do the surveys, everyone. Thank you for attending. Are there any questions here? Thank you for attending. Are there any other questions? If anybody wanted to unmute, if they had a question, and ask it or raise a hand. Oh, we've got another one in the room.
I do have one other question
Yeah.
about if there's been any discussion about kind of like artificially generated like electronic sounds versus kind of I don't know, sort of natural
Yeah.
sounds. Has there been any discussion or talk about what those differences are?
Yes, there is, it's kind of there's definitely, it's where everything's headed. And so, yeah, these were using pure tone, very clean sounds, but in the wild, that is not the case. And so what a lot of the research is showing is that we're using this foundation of audiograms, and we've got this idea of the spectrums that they hear and how quiet they hear. So there's a lot of that picture that's been filled in, but it's not necessarily natural. So that's a huge place that everything's going. And that's kind of the same connecting to just noticeable difference is it's this very small difference in how they're hearing, but it does assess a totally different aspect of what the ear is perceiving rather than just a single tone, like a difference. And so within those, like within the sounds, there's so much going on. So that's a huge place. I didn't talk a lot about it because it's really complicated, but that's the direction that things are going in. Some of the research is showing that when you start to use a more complex sounds, the models that we had, that we were based on the data we have, aren't predicting as accurately as they should. So they're to give you a real simple version of some of the ones that I've read. Yeah.
I have another question, and then maybe just the committee also more questions. But so if we had a soundproof room,
An anechoic chamber. Yeah.
that would be, yes, one of those. If we had that, how difficult would it be to train these animals, especially around the lip, to just signal that he hears something?
So essentially to do an audiogram.
Yes.
I think it would be easier than training a just noticeable difference task
Yeah.
personally. Yeah,
So we just need one of those.
we just
here.
need one of those.
Okay. That's what the NSF is for. so.
Yeah. Great, lots of good questions. Thank you,
Any other
guys.
questions?
I don't know.
All
Okay.
right, another round of applause.
Well, thank you guys so much. I think this is the point at which we do a closed session.
Yep, now we're gonna do a closed session and talk to the committee
So
members. but
Jason and
Neil,
Jason and Dorian stick around.
And we will see you later. The mask.
I knew I had one somewhere.
Okay.
Hopefully I can get rid of it too.
Yeah.
Thank you for all you're doing.
Yeah, well, we're gonna be working on it, so,
We appreciate it.
yeah, the Pro World is a great resource for the community here. So, all right. See, I like your shirt.
Yeah, we have to stop matching when we leave the house.
Yeah, coordinating.
Right.
Alrighty.
Okay, so I've never used this. I was trying to get Zoom on my phone.
All right, let's see. I guess I'm gonna say goodbye to Trudy and Suzanne at this time. Thank you guys so much for coming.